In this lecture, we're going to go over the cell cycle, which is how cells do their job and divide. So the cell cycle is essentially just a repeating sequence of cellular growth and division that occurs during the life of an organism. The cell cycle is made up of several phases. So the first one we'll talk about is the G1 phase. This is often the longest portion of a cell's life. During this time, the cell grows and carries out routine functions, and cells that are not dividing remain in this phase. And the G1 phase is part of a larger category called interphase. And interphase makes up the G1 phase, the S phase, and the G2 phase. Some cells remain in the G1 phase forever, like muscle cells and nerve cells in adults don't divide, and so they just remain in the G1 phase, and if they die, the body does not replace them. If a cell is going to divide, then it also goes through the S phase, which comes after the G1 phase. So during this phase, a cell's DNA is copied, and at the end of this phase, each chromosome has been doubled so that it now consists of two sister chromatids that are attached to each other at a structure called the centromere. After the S phase comes the G2 phase, and here we have preparation for the division of the nucleus occurring. And so microtubules are a structure that are going to be important later on in cell division, and they are assembled, and the DNA is already replicated, so now we're almost ready for the cell to divide. Before we talk about how a cell divides during mitosis, we'll talk quickly about cancer. Cancer is essentially a disease that is defined by unregulated cell growth. So if cells divide more rapidly than they should, they can grow into structures called tumors, and they can cause problems if they steal too many nutrients from cells that are needed to survive. Next, we're going to talk about mitosis. So remember, the G1 phase, the S phase, and the G2 phase make up interphase. After interphase occurs, when a cell divides, that process is called mitosis. So mitosis is the division of the nucleus and then the cytoplasm, and that results in two identical daughter cells. There are four major phases or steps that occur during mitosis, and they are called prophase, metaphase, anaphase, and telophase. During prophase, the first phase of mitosis, chromatin, which is made of DNA, shortens and thickens into chromosomes so you can see them more easily. So here you can see the DNA is becoming thicker strands and more visible. Another thing that happens is that the nuclear membrane, which is the membrane that goes around the nucleus, disappears and disintegrates. And then there's structures called centrioles that move to opposite ends of the cell and send out a structure called a spindle, which will be important later when we're dividing up the DNA between the two new cells. After prophase is something called metaphase. It's the shortest phase of mitosis, and it occurs when the chromosomes that are now highly visible, you can see them here, and there's in the cell there's 1, 2, 3, 4 chromosomes, and you can see that they line up along the middle of the cell, and you can see that that spindle structure that we talked about during prophase has been formed, and so it's these little strings that attach to the chromosomes, and they were produced by these structures called the centrioles. And the centrioles are on opposite ends of the cells so that they can pull those two halves of the chromosomes away from each other. During anaphase, the centromere, which is that structure that was holding the two sister chromatids together, is pulled, and one chromatid from each pair is pulled to each end of the cell. And so those two sister chromatids are now pulled apart so that we have 1, 2, 3, 4 going to this end of the cell, and 1, 2, 3, 4 going to this end of the cell. So that divides the DNA in half evenly. The next phase is called telophase, and this is the reverse of prophase essentially. So this is when the cell membrane, DNA starts to become chromatin again, so it spreads out and the strands get thinner and things like that, and then the cell will start to pinch off. When the cell pinches off, kind of during or after telophase, that process is called cytokinesis, and there are little structures called microfilaments, and you can see them easily in this illustration. those microfilaments are going to pinch off the cell and eventually cause it to form two new cells that are completely independent of each other. And both of those two new cells have a nucleus, so you can see it's formed there, and then it's present in the two new cells. And that nucleus is going to have identical copies of the DNA because the sister chromatids were pulled to both sides of the cell to fill each of those nuclei and make them identical. In plant cells, because they have a cell wall, cytokinesis looks a little bit different. The cell can't pinch off because the cell wall is rigid. So instead, vesicles that contain cell wall material deliver cell wall material to the center of the cell, and then from that material they can build a plate, and eventually the plate goes all the way to the ends, and we end up with 2 new daughter cells. So to review the whole process, remember a cell that's in the cell cycle, before it does mitosis, will be an interphase. Interphase includes the G1 phase, S phase, and G2 phase. When a cell divides, it begins this process called mitosis. And so remember, during mitosis, first the DNA becomes visible and the cell membrane starts to disappear, and then the chromosomes during metaphase line up along the equator of the cell. During anaphase, those sister chromatids are pulled apart from each other, and then during telophase, the cell pinches off so that you have 1 and 2 new cells. This process is a little bit simpler in prokaryotic cells because they don't have a nucleus. So prokaryotes always reproduce asexually, so that's when a single parent passes on exact copies of DNA to all of its offspring. And these cells always go from 1 cell to 2 daughter cells. And the name of this process in bacteria is called binary fission, and it produces clones or identical offspring. So here's a diagram showing how that works, right? So here we've got a cell, we've already made a copy of the DNA because here's 1 copy of the DNA, and then here's the other copy. So those copies are identical. Those are going to go to the 2 new offspring. So once the DNA has been replicated, all you have to do is pinch the cell off. So you can see that furrow is starting here, and then here the cells are becoming more independent, and now we have 2 completely separate cells, and those are the 2 daughter cells from the original parent cell. Bacterial cells do not undergo mitosis because they do not have a nucleus, because mitosis specifically refers to the division of the nucleus when a cell is dividing. And lastly, these are what cells look like in real life. So these are microscope images of cells in the various phases of mitosis and interphase before mitosis begins. So you can see this cell here, for example, is an interphase. It has a very well-defined nucleus, and you cannot see the individual chromosomes inside of the nucleus. If you look at this cell, and so this is the plant cell, right? It's got cell wall. These are onion root tip cells. Here you can see the DNA is starting to become a little bit visible inside the nucleus, but it's still not extremely well defined. And then in the nucleus of this cell, the DNA is even more visible. And then by the time you get to this cell, the DNA is quite visible as chromosomes. When you get to the 6th cell, so here's the entire cell, and you can see that the chromosomes are all lined up across the middle of the equator. So that is metaphase. And then in this cell, you can see the cell chromosomes are starting to get pulled. Eventually they're going to get pulled to opposite ends of the cell. You can see that more clearly in cell number 8, how those cells or those chromosomes are getting pulled to opposite ends. And then in number 9, this is telophase. This DNA is clearly at one end of the cell. The DNA here is at the other end. Those are eventually going to be enclosed in the new nuclei that you can see in cell 10, which is the end of telophase. And now you can see we actually have 2 new daughter cells. So there's 1 cell here and 1 cell here.